We're going to pick up where I left off on the previous video. We were talking about the different kinds of media. We did the physical state, then we did the chemical composition, whether it was synthetic, very well defined, or if it was complex, not quite so defined, but nice and rich. Now we're going to classify the media that we use by its functionality and how we use it. And one of the types that we use is called enriched. We use that an awful lot. When we first take a sample for some place, we often put it on enriched so that it has a good chance of growing because we want to take a look at it. We're certainly going to use it on bacteria that we refer to as being fastidious. And fastidious, as you see here on my slide, is defined as being picky, which means essentially there are lots of nutritional requirements that they have. And if you think about it, if we got a pathogen growing in us, they're using us as their petri dish, so to speak. And we're pretty much a complex media, and they need all the things that we have to offer them. So using an enriched media to grow a sample is often a good idea. Some examples of enriched media. This one here is a blood agar. This particular image doesn't show the blood as red as it normally is. This plate's usually bright red. But after you've incubated it for a while and the bacteria's had a chance to grow, it may turn a little bit darker. This top one up here is something like tryptic soy agar, where they've taken soy protein and digested it with enzymes to make it a little easier for the bacteria to use. This one here is actually called chocolate agar. In this case, they put the red blood cells into the agar before they melted it, and that causes the red blood cells to burst open and it changes the color. Whereas this blood agar, they put the blood in after it's been sterilized and it's almost getting ready to set and harden. That's when they add the blood. So there's a little bit of a difference. Another type of functional is called selective media. And this is where we are going to use a particular agar to enhance the growth of one bacteria but prevent the growth of some other kind. And there can be any number of selective agents in here. I listed high salt. If we're trying to isolate staph, which grows on our skin a little bit saltier than other parts of our body, you could think of it that way. And so it does grow at higher salt, but that inhibits the growth of a lot of other bacteria. We have bacteria growing in our intestinal tract, and they're used to seeing bile salts. It doesn't bother them. But something like our staph might be inhibited by it. In other cases, we might be using antibiotics. When they came across Legionella way back in the 80s or 70s, it was mixed. It tended to be mixed in with a lot of other bacteria, and they ended up having to use antibiotics that it was resistant to naturally to help isolate it. Now, we also have a tendency to mix selective media with differential components, and that's obvious, especially in this bottom one. You can see some of the colonies here have this red color, and these others have this purple color, and that's due to something that we have in the agar that is giving us a difference in the two different kinds of colonies. You can see on this one, this is actually selecting for Pseudomonas that's producing this nice green pigment. I've listed some of the others that are available. You don't have to really learn about them, just so you could see. I mentioned the mannitol salt agar. We're going to talk about it in a little bit. We have one for isolating fungus, but bacteria is not going to grow on it. Depends on what we're looking for, and we can usually isolate it. So I alluded to this in this previous slide, that we can add components that will give us a difference between different bacteria genera. And using these various differential components, we may end up with a different colony color like we saw. We can also look to see if it has different enzymatic capabilities. And this particular table right here shows you some differences. Like blood agar has red blood cells in it, and what that's going to do is it's going to allow us to see if the bacteria is able to lyse the red blood cells. A good example of using this is for somebody that we suspect has strep throat. Uh, the, when they swab the back of your throat, they put it on blood agar, and we get these little colonies growing, but they have this clear zone around it because the Streptococcus pyogenes, which is what's causing the strep throat, is lysing the red blood cells, and it makes it clear around it. So it's a very easy way of differentiating Streptococcus pyogenes from some other organism or virus that's giving you a sore throat. So you can see there's a number of different ones that are available as well. Again, we don't always use it in a plate. Here is something that's called the triple sugar iron agar that tells us some differences between bacteria. This is uninoculated. This particular bacteria is able to utilize one of the sugars, but not the other two. This one's using all three of the sugars, and it's producing acid. And this one's actually taking the iron that we have in there, and it's precipitating it and giving you this black color. So we use pH indicators that tells us when acid's being produced. There can be other dyes. Here's a really good example, this chrome agar. You can see they've streaked E. coli, Klebsiella, Proteus, Enterococcus, these different ones, and you can see they all give you different colors based on how they're reacting with the dyes that are in the agar. So we do tend to use differential and selective together. It's kind of like using one plate to tell us a number of different things. One of the classic examples is this mannitol salt agar. The name tells you what's in it. It's got mannitol, which happens to be a sugar. A lot of bacteria will take sugars, ferment them, and in this process of fermenting them, they produce an acid rather than an alcohol. Many of you might be thinking, well, fermentation means it's producing ethanol, but with bacteria, they're actually producing an acid. And they put a pH indicator in here. So if the organism is able to use the mannitol and produce the acid, it changes the color. So you can see this area right here where nothing's growing. That's what the plate really, really looks like. But here we have Staph aureus, which is one of the an organism that causes skin diseases and other things. You probably have heard of MRSA, which is a Staphylococcus aureus that's gotten very resistant to bacteria. But here it's producing acid from the mannitol, and it changes the color of the agar. Now on our skin we have the Staphylococcus epidermidis. It grows on the high salt, but it doesn't ferment mannitol. So if we've got a skin wound and we take a swab from it, there's a good chance that you're going to get Staph epidermidis in there just because you have it on your skin. But there's Staph aureus on here. We're going to be able to see the difference on the plate. And this 7.5% sodium chloride is a fairly high salt concentration. If you think about it, when you get an IV in the hospital or wherever, emergency room, that kind of thing, that's at 0.9% sodium chloride. So this is quite a bit higher. And so it inhibits a lot of bacteria, including the gram-negative. So we use this for isolating those. On the other hand, we might be using McConkey's agar if we're looking for gram-negative bacteria and we want to inhibit the gram-positive. This one's got multiple parts to it. Lactose, which is a different kind of sugar, so it takes different enzymes to deal with it. Again, when it ferments the lactose, it produces an acid. We can see that here. This particular colony here is utilizing the lactose. It produces this pigment, or it produces acid and causes this nice bright pink. When it's not fermenting lactose, we don't get that pink color at all. It has biosalts in it, which inhibits the gram-positive, but allows lots of gram-negatives. And this is kind of showing you what's going on. Here we have E. coli, which we know we have in our intestinal tract. Really strong lactose fermenter. Gives you this nice, looks more like red, but it's really a pink. Enterobacter aerogenes is a gram-negative. It can grow. As well as both of these, but Proteus and Salmonella, neither one of them ferments lactose. So they grow on it, but they don't change the color of the media. And here's Staph aureus, our gram-positive, not able to grow at all. So it's quite helpful for us. This little diagram here kind of gives you a better idea of what's going on. Let's say we have a mixed sample. It could be a fecal sample. It could be a swab from a wound. Any kind of sample that the doctor wants to be taken from the patient. Well, then they put it on a selective media if they suspect, okay, we have this sputum sample, and we suspect that the person may have tuberculosis. So we're going to put it on this particular media that selects for tuberculosis. But we could also be another kind of organism that's causing the lung problem, so we're going to put it on this kind of media too. But just in case, we're going to put it onto an enriched agar to give a chance for everything to grow, so that if nothing grows on the selected media, we still have a sample. Again, same kind of thing on the differential. Again, you would plate it on a differential media. You may have more than one kind of thing growing, but different colors. And again, oftentimes in the lab, we have selective and differential combined together. So this is a summary of the media types we're talking about. If we're talking about physical state, we have the solid, very useful, used a lot. We have liquid, used for biochemical tests or maybe growing large amounts in the biotech field. And then we have the semi-solid, which tends to be for specific tests that tell us something about the bacteria. If we talk about media types based on their chemical makeup, we have the synthetic. We often use this when we're growing photosynthetic bacteria. I'll give them a very defined one. Or we have complex, where it's got lots of rich media and we're not 100% sure what's exactly in there. And then we can classify things on its functionality, whether it's enriched, or pretty much everything we think is going to grow, or differential selective. And here's a different differential selective one here. You can see it's got these little halos. It's black. That tells us something about the particular thing. So those are the different types of media